Bismillahirrahmanirrahim. My name is Dr. Awais Masud. I am working as Deputy Director Livestock in Livestock and Dairy Development Department, Punjab. Nowadays, I am doing my MPhil in microbiology in University of Veterinary and Animal Sciences, US, Lahore. Today, I want to present my research work probing the anti-diabetic effect of Osimum sanctum lin holy basil leaf extract on alloxan-induced diabetic rabbits. As we all know that diabetes mellitus is a worldwide problem. It has two types, type 1 and type 2. Type 1, which is juvenile onset diabetes, is permanent and it involves usually uh, damaging of islets of Langerhans, while type 2 is usually considered as due to environmental factors. WHO says that it is increasing day by day, and Pakistan is at 7th position out of 10 highest prevalent countries. Uh, we know all that medicinal plants have very importance in treatment of different type of diseases. Among them is holy basil, which is also called Osimum sanctum leaf plant, which has quite importance in treatment of different types of disease conditions. This plant in Pakistan is usually known as tulsi or also called as niazbo. It is usually known by its seeds, which are toham malanga, which are being used in summer season. Holy basil leaves have a sacred value in India, and in America, it is declared as generally recognized as safe GRAS plant by FDA. This plant is very important in treatment of different types of diseases. Uh, I retrieved the idea of its effect on diabetes from different types of ukma. In Ayurvedic or Hakim, use this plant for treatment of anti-diabetes. So my study is somewhat a preliminary type of study, which involves scientifically inspection of the plant for use of for diabetes. This study involves the use of rabbits, which are lab animals. We prepared ethanolic extracts of polybasil leaves and we did phytochemical analysis, detail of which will come in next slides. We did animal trial. We took 25 rabbits and made 5 groups, having 5 rabbits in each. T node was control group, which was first group, and it was given on a placebo feed or normal feed, while other 4 groups, T1, T2, T3, and T4, were induced by diabetes by a diabetogenic agent, which is called alloxan. After 72 hours, we did blood glucose test for confirmation of diabetes. As all we know that diabetes have two major roles, which are weight loss, continuous weight loss, and increase of blood glucose. So for monitoring of these two parameters, we conducted 4-week study, and each and every week we did these two tests. While we did complete blood counting, liver function test, and renal function test at start of experiment before the injection of alloxan, and at end of experiment at when doses were completed. To T naught with this graph, table shows you that healthy animals were given normal diet, while T1 group was also given normal diet, while T2, T3, and T4. These all are were given alloxan to induce diabetes. So these were diabetic rabbits. These were given 100 milligram, 200 milligram, and 300 milligram doses of holy basil leaf extract. Uh, in results, you can see phytochemical analysis, and you can see that total flavonoids are much more in the olive leaves, while antioxidant activity is much higher, which makes this plant useful for different types of disease conditions, for treatment of different types of disease conditions. Uh, we also can see that in our study, we observed that while increasing amount of this holy basil leaf extract, we gradually have controlled blood glucose level, while also weight of loss was minimal. So 300 milligram dose have much more effect, significant effect than other doses. As I already discussed that FPG and weight loss were measured in weekly basis, and we observed that 300 milligram dose gave maximum result. CPC before and after when were compared, we see that monocytes, lymphocytes, and ESR were increased while granulocytes were decreased somewhat. While LFT shows ALT was somewhat increased, while liver kidney function test showed that blood urea level was increased after treatment. Pre-treatment values were down. If I conclude my experiment, I would like to say that these polyvagal leaves can be used for treatment of different. I conclude my study that polybasal leaves, when used for treatment of diabetes, they decrease blood glucose level and also minimize the weight loss. Uh, Obviously, by increasing the dose rate, they have more effect. Our biochemical tests like LFT and RFT showed that they have minimum toxic effect on liver and kidney, while hematology also showed the same sequence. So in my opinion, to establish pharmacological significance of polybasal leaves, uh, further investigations for dose rate and safety margins are necessary. Thanks for patience. Allah Hafiz.